This is a production of Cornell University.
Well, thanks a lot for this nice introduction, and it's really a great pleasure to be here with, in the back, a nice poster of Barbara McClintock. So the place to be, and there's always a kind of battle, and I always also see these numbers, who is the #1 university in the world. Well, I think we should not discuss that at all. I think it's nice to see that there are a couple of centers in the world that do good plant science and agricultural research, and for sure Cornell is one of them, and I think also Wageningen is a nice center where a lot of different disciplines come together. So as introduced, we work on a lot of different species in my group. A little bit of work in tomato, quite a lot in cauliflower. Since 2012, also a lot in bulbous plant species, mainly focusing on tulip. But for the majority of understanding the underlying molecular mechanisms that I will talk about today, we still do that in Arabidopsis. So for those who are coming, especially for work in crops, I have to disappoint them. But there are a few steps towards cauliflower and also one small step to tomato. So I don't know who of you have ever been at Wageningen University. Well, in one thing you have beaten us. Your campus is bigger, nicer, much better, I would say. But we have also a new campus, and that's what you see over here, with 2 major education buildings, and it's all quite new. It's I think about 10 years ago now that we moved from old buildings within Wageningen to a campus just outside of Wageningen. And it's nice and compact, and we have about 12,000 students nowadays. And here you see it from the other side. And what is a good thing of it, so Wageningen itself is just here on the side. So it's directly adjacent to Wageningen. It's not only university buildings, but this one is, for example, the dairy company, the research department of Friesland Campina. So there's a lot of integration also with spin-off companies. So this whole building is full of spin-off companies, but also big players in the food and agricultural industry. And you see here this grass field in between. At this moment, Unilever, the big food company, decided to combine all their research in Wageningen. So at this moment, they are making a huge building here, actually filling up the whole space between these 2 buildings. And there will come about 500 researchers very soon start there. So the whole Unilever research will all be at the campus, which gives a lot of opportunities for cooperations directly with the industry. Well, this is the plant science feeling with the can nicely recognize it from the green sun screens. Of course, only on sunny days, otherwise it's just glass that you see. And in the back you see the greenhouses, and also here some research fields where we do the experiments with the field crops. So what I will talk today about is plant adaptation, and just to start off, in the end it will all be Arabidopsis, but I think I don't have to tell that here, but often I have to convince people why plants are so interesting. And one thing that is really remarkable of plants is their tremendous capacity to adapt to all different kind of environments, as seen here on this picture. And it's also needed for plants because plants are sessile organisms, and well, when it's cold, we walk inside and protect us in that way from cold temperatures. But plants have to find a way to survive under all different kind of conditions that they don't have any influence on, and they just have to take it as it comes. Well, they are so successive in conquering the world. You also see that here you have, of course, optimal conditions, high humidity, a lot of nutrients, high temperatures, but you see there's also then a lot of competition. So what makes plants so successful is they adapt to their specific environments, and it can either be very harsh environments, but the good thing is there's less competition, or very rich environments, but then they find ways to compete with other plant species. Well, if you want to adapt your whole growth cycle, way of living, structure to the environment, you need ways to measure the environment. And there are, of course, a lot of different signals, nutrient levels, day length, light quality, but also temperature is a very important signal. And in our lab, we focus completely on how plants, and in this case Arabidopsis, is able to adapt its whole growth to temperature. Well, in this respect, you have to realize that there are 2 major differences in temperature. You have the more extremes that can either be stressful temperatures, but also important temperatures. And for that, a very nice example is vernalization. That means winter cold. So Arabidopsis accessions coming from northern countries, they need, in order to flower, a long period of winter cold. So that's to prevent that they will not set seed in the autumn. But they need a long period of cold, and only after that long period of cold and forming a lot of rosette leaves, they are able to flower. So this is called the vernalization response. So quite extreme. Another example of that is breakage of dormancy, what you have in a lot of trees. So this bud breaking. The buds are, you see it nicely now outside. We see all these fruit trees starting to flower. These buds need cold in order to open up and to flower. Besides that, there is the ambient temperature response, and that's much less studied in general. There's just the daily fluctuations in temperature. And a lot of people think, well, one day it's warm, the other day it's a little bit colder. It doesn't matter that much. But plants are measuring that very precisely and change their development based on the temperature they measure. And that here you see one of those phenotypes that is in growth response. So if you go to a slightly higher temperature, you see that in Arabidopsis the petioles start to stretch, and also the leaves are standing more straight up. And this is in this case, you can imagine that you can have more aeration. So the plant has far more cooling capacity at higher temperatures due to this change in its morphology. Well, we do a little bit on those growth aspects, but the main biological process we are interested in is flowering time. And that is also heavily responding to temperature. The vernalization response, but also to ambient temperature. And that's what you see here when you grow Columbia at different ambient temperatures. Well, here is blue, here red, but this means colder, this means warmer. But even a 2 degrees difference in average temperature can already speed up the flowering, can double the speed for flowering. So it has a tremendous effect on flowering time in a particular range. Another reproductive trait that is very much influenced by temperature is seed dormancy. So seeds, after they are produced, depending on the temperature, they can also moisture level and a lot of other signals, they decide to germinate or not. But also their level of dormancy that they have at the moment they are formed, it's well known that that depends not only on the temperature that the germination does not only depend on the temperature they are experiencing when they are already a seed, but also the temperature that is experienced by the plant before or during the seed set, that is determining the level of dormancy. And it was already long known that during flowering, the temperature then, so at the moment that seeds are developed and the embryo is developing, temperature matters, but quite groundbreaking work of the Steve Penfield lab at the John Innes Institute at the UK, they showed that Arabidopsis plants that are exposed to cold temperatures during the vegetative stage and then move to warm temperatures as soon as it starts bolting. So during seed set, it's warm temperatures. The seeds that you harvest from those plants are more dormant. That means that it's not only the temperature that the seeds experience themselves, but also the temperature that the plant is experiencing that is taken into account for seed dormancy. And it also means that there must be memory. So there must be a kind of memory of this temperature. Well, plants don't have brains, and then it's of course interesting how does such a memory work. I will come back to that later on. So what you see here is, so I just showed you that Arabidopsis columbia gets earlier flowering, but in this response, there's a lot of variation. So here you see flowering time of all kind of Arabidopsis accessions. When you compare 27 to 22 degrees, and you see nicely that Columbia flowers earlier. And you see that the majority of accessions flower earlier, some far more extreme than Columbia. But as you can see, there are also accessions that when you increase temperature, they decide to flower later. So you see this whole range of responses depending on ambient temperature. And that you also see back in crops. So cauliflower, also a Brassicaceae, very close to Arabidopsis in that respect, from the same family. But it responds different than Columbia. So if you go to a higher temperature, then the induction of flowering, which is the induction of curd formation, which is one big inflorescence, gets delayed. So a slightly increase in the ambient temperature delays curd formation. And it's not only delaying curd formation, it's also affecting then the outgrowth of the inflorescence, so of the curd. You get a more open curd due to more stretching of the inflorescence stems. So temperature has a clear-cut effect on also on our crops, and small fluctuations in ambient temperature can affect a lot when we can harvest our crop and what will be the quality of the harvestable product. Well, then, of course, a question that we have to answer if you want to study temperature: how do plants measure temperature? Do they have a kind of thermometer somewhere in their structures? And for mammalians, that is quite well known, and that's maybe also not so strange, that there are membrane proteins. And membranes, I think we all learned during our physical classes that their fluidity is depending on temperature. So you could imagine that there are membrane proteins that can act as thermometers. But if people looked based on homology to this kind of proteins that are then in the membrane and sense the temperature in mammals. They were not able to find anything that looks alike in plants. It does not mean that there is not a membrane protein, but at least these classical temperature sensors, they are not there. Well, then is of course the question, what is then measuring temperature? And then once more, I think it's good to realize if you look to, do you really need a temperature sensor? Or is it maybe inherent to all the molecules? And the latter is the case because we all learned that folding of proteins, looping of RNAs, all that is temperature sensitive. So maybe there is not a general temperature sensor in plants, but just the molecules that play a role in growth and flowering themselves by their stability, folding, protein interactions, all that kind of characteristics can sense temperature. And we more and more start to believe that that is the way how plants sense temperature. Nice examples of that are a recent paper where they showed that phytochromes, phytochrome B, which is known to change its way of structure under influence of red or far red light. Its structure, its stoichiometry can also change under temperature. So phytochrome B is the major light signal receptor, but it can also act as temperature sensor by changing its whole folding upon a difference in ambient temperature. Another process that is heavily temperature dependent is gene splicing. So in eukaryotic genomes, there are, in the majority of genes, there are introns. And depending on how these introns are excised, you can get different final transcripts, mature transcripts, encoding for different proteins. And this gene splicing appears also to be highly temperature sensitive. And that is what we focused on over the last 5 years and where I will show the most of. So when we started this project, it was already known that splicing is heavily affected by temperature. And then we thought, well, can we get more genome-wide insight in which genes undergo differentially alternative splicing? And you will see this term, DASE. You will see it back a lot on my slides, which means differentially alternative splicing. And which transcripts are now all sensitive for their splicing on temperature. So what we did is we have been growing Arabidopsis plants at 16 degrees under short day conditions. So that's not flowering inducting conditions. And then when you grow them for 5 weeks, they are still perfectly vegetative. We also tested that by looking to leafy and apetla1, so floral meristem markers. And then what we did is after 5 weeks, we switched half of the plants to 25 degrees. So still ambient, so it's not stress temperature. Arabidopsis grows very well at 25. And then after exactly 1 day, 3 days and 5 days, we took samples of the meristem enriched, so the meristem plus the young leaves. And from that we did RNA-seq and then de novo assembly to see all the new splicing variants that maybe are going to appear. We decided to take exactly 24 hours later, 3 and 5 days, to have no influence of the clock, because it's very well known that clock genes get also differentially alternatively spliced. So if you then start sampling after 4, 6 hours and so on, it's very difficult to figure out what is ongoing. So that's the reason why we took the first sample after exactly 24 hours. Well, to summarize that, if we then look to which genes are differentially alternative spliced, they are here. You cannot read anything there, but it's about 1,000 genes. We'll zoom in later on the type of genes. So it's definitely something that is not just missplicing because of a higher temperature, because then you would expect that the majority of genes are differentially alternative spliced. It's a specific set, and I will show you there are also specific classes of genes in that set that are more sensitive for this temperature splicing. What you also see is that when you compare 16 to the 25 samples, the biggest difference is between 16 and 25, day 1. There you see a lot of differences, and then it directly more or less stabilizes. So it's a very fast response, but if you keep the plants at the warm temperature, they also keep this altered splicing ratio. Only when you place them back to low temperature, they swap again. So the conclusions here were many genes undergo differential alternative splicing, but there seems to be some specificity. It's not just simply missplicing. And it's a fast response. Most changes occur in the first 24 hours. Well, what are then the genes that are differentially alternatively spliced? Well, if we look to overrepresented gene ontology terms, which was not surprising, we find back flowering. And we knew that flowering is heavily temperature dependent. And so we see that many of the flowering time genes undergo differentially alternative splicing. So this was as expected. But what we didn't expect at that moment was that all the top classes are RNA splicing, mRNA splicing, RNA metabolism, RNA processing. So genes involved in splicing itself. So that makes it complicated, because then you start to think, how is that possible? Alternative splicing is changing, but the genes that are needed for alternative splicing, they are the most heavily differentially spliced when you change temperature. Well, probably have all a view on how splicing works from your textbook knowledge. For me, that was the same when we started this work, and I knew that splicing was a complex thing with many proteins involved, but I didn't realize how complex it is. Nowadays, there are almost 400 proteins known that are needed in this whole splicing machinery. So it's not a very simple cutting of introns out. It's a very complex machinery of and set of genes that are involved in this. And when we started our work, it was already known that genes encoding for so-called SR proteins, arginine serine-rich proteins that are in the core splicing machinery, that they are alternatively spliced. But what we realized is that actually all the different types of genes that are involved in splicing, many of them get differentially spliced when you change temperature. So temperature has a very strong effect on the whole splicing machinery at all different kind of levels. Well, based on that, we came to this model, and what you see is that when you change ambient temperature, you get a very fast response, especially in the splicing-related genes. That's an initial response. As a consequence of that, you get an altered splicing machinery, altered complex of the splicing machinery proteins. And that then, as a second boost, can act on developmental genes, which then gets differentially spliced, leading finally to adaptation of the phenotype to temperature, for example, late flowering or stretching of these petioles. Well, this is a model, but a current working model that we have. A question then, of course, is what are then these developmental genes that are specifically differentially spliced by this altered splicing machinery? Because in the end, that are the genes that are important to change the traits of the plants. So when we look at that, this is just a small example list. You see here different genes, so flowering locus M, also known as METs affecting flowering, MAF3, MAF2, PRR, so the clock proteins. Here you see the splicing event. RE means intron retention, exon skipping, alternative 5, alternative 3, mutually exclusive exons. So you see also that temperature affects all these different types of splicing. It's not that only intron retention or whatever is affected. All the different types are affected. And then here you see the splicing ratios, particular isoforms, 16 degrees, 25 degrees. And well, you see that in some cases the effects are quite big, but you also see that sometimes the effects are quite mild. It's significant, but it's not that in once you change from isoform 1 to isoform 2. It's often a small switch in their ratio. Another important thing is what you see is that it's quite stable, so that's indicated here. So this means that at 1 day, 3 days, and 5 days, we saw a significant difference in comparison to 16. And you see that the majority of effects is very stable. When you change temperature, it's changing and it stays like that. Well, this is a selection, but we see many of these MAF-like proteins, and then it's the question, what are that for a kind of proteins that are then alternatively spliced, or the genes encoding them? And these MAF-like genes, they all belong to the METS domain transcription factor family. And well, it was already mentioned in the introduction, our lab is mainly focusing on transcription factors. So I was very happy to see these candidate genes, and that's also why we focused on these genes. That does not mean that other genes are not differentially spliced, but we focused on transcription factors. And these MAF ones, they are quite interesting because the major gene in the vernalization response in Arabidopsis is FLC. So this is the gene responding to the winter cold. And all these others here that are close relatives of FLC due to duplications of the FLC locus, MAF1, MAF2, MAF3, 3 of them undergo differentially alternative splicing. So it seems that both cold and warm temperatures heavily act on this FLC clade members and in that way change flowering time. So more in depth, there was already at that moment genetic data from the lab of Detlef Weigel in Germany that especially FLM is a major player, because if here you see again the thermosensitivity, so the change in flowering time when you change from 22 to 27 degrees. So you see that's almost twofold change in that it flowers faster. And in FLM mutant, this effect is almost completely gone. So you see a ratio of 1, meaning that it flowers at the same moment at low ambient temperature and high ambient temperature. So FLM is a major player, but also his sisters, brothers, how you want to call it, these other duplicates, they have a clear-cut effect. That's what you see here. Here you see a MAF2 mutant. So normally, again, wild type, 21 degrees early flowering, 16 degrees late flowering, and you see that at late flowering, the MAF2 mutant is much less. It flowers Not exactly at the same moment, but you see already a quite clear-cut effect. And that's also what you see back here. Here you see the MUF2, the two major isoforms, variant 1 and variant 2. And you see at 16 degrees, you see mainly the small one. And when you go to 27 degrees, you see mainly the top one. But it's not black and white again. Another one, MUF3, that's more complicated. This is data from an RNA-seq from our own lab. And what you can see there is that there are even 6 different isoforms. And when you change from 23 to 16, you see again that this bottom one here is disappearing. Some other ones are getting more intense. So you see a difference in the ratio of these isoforms. Well, what you can see also here for MUF3, we don't have Newton data to show that this is really important for the flowering time. But one thing that is also good to realize, this was based on RNA-seq and de novo assembly. From all these isoforms that we identified, so this was the RNA-seq, there we identified them, then by qPCR we could confirm. We have cut all these bands out and sequenced it, and it were exactly these isoforms which were predicted by the bioinformatician, but only 2 of them are available in TER. So that also shows that splicing, a lot of that information, if you want to study it, yeah, looking at the TER database, although it's a very rich resource, there is a lot of information still missing. And there are definitely a lot of novel isoforms that are missed in these databases. Well, then the question is, how can 2 different transcripts from the same gene with a small difference in it, sometimes only a very small extension somewhere in the middle of the protein, have such a different function in flowering time? So that was the next question we asked, and for that we initially focused on FLM. So FLM is one of these repressors, and it's the major one. That's when you have a knockout, is almost completely temperature insensitive. And if you look to FLM, you find in Colombia mainly 2 isoforms, beta and delta. And FLM has 9 exons, so it's a quite complicated gene, but you will never find a transcript in which all 9 exons are present. So it has a so-called mutually exclusive exon. So you find either exon 2 in the transcript, and then we call it beta, or you find exon 3, then we call it delta. Well, at that moment, we did, together with the lab of Marcus Smith, overexpression of these different isoforms. And if you overexpress the isoform, which is the most pronounced at cold temperature, you get a late flowering plant. Whereas when you overexpress the delta variant, which is the most pronounced at high temperatures, you get an early flowering. So confirming that the switch in isoform intrinsic into that protein, by changing a small piece in the middle of the protein, you change its functioning from a repressor of flowering into an activator of flowering. And that's what is summarized here. So at low temperatures, you have mainly beta, so mainly the repressor, you get a late flowering plant, whereas at high temperature, mainly the activator, and you get an early flowering plant. Well, we are molecular biologists, and then we were not satisfied by this. We also said, what is then the underlying mechanism? How can 2 proteins that are very similar do a complete different function? So for that, we did a bunch of experiments, mainly in vitro experiments, DNA binding, protein-protein interaction studies, and it's already all published quite some time ago, so I will only show the final summary of that. But what we could figure out that at a low ambient temperature, you have mainly FLM beta, and FLM beta is a transcription factor that needs to dimerize with SVP, another matchbox transcription factor. Then they can bind as a complex that we've shown by chip-seq experiments to, for example, the FT promoter, the SOC promoter, so floral activators, and they repress its expression. When you go into higher ambient temperature, you get less of beta, you get more of delta. Delta can also form complexes with SVP, but these complexes cannot bind DNA anymore. And in that way, you get competition between these kind of complexes, which are in principle non-functional. So they don't repress the floral activator anymore, and these repressive complexes. And in that way, 2 proteins that are very similar by binding to the same interaction partner, SVP, give their function. Well, I mentioned already, this is mainly based on in vitro work and on chip-seq experiments. After this publication, a lot of discussion came, is this model correct? And there was work from Suraj Balmas, Romanian. He's now in Australia. There's this paper in Nature Plants. And what they say, actually this model is not correct, because FLM delta, the transcript that we show with the mutually exclusive 3rd exon, according to his lab, that is not existing. And what is existing is a transcript with an intron retention. So you get less beta, but you get more of a transcript with intron retention. And these transcripts are targeted for NMD, nonsense-mediated RNA decay. So what is happening at a low ambient temperature, you have a lot of beta, that gives a functional protein doing its job, but at a higher ambient temperature, beta is going down, but you have a lot of delta, but it's never making a protein, it just gets destroyed. So still the splicing is important, but the delta variant is not leading to a functional protein, it's leading to a transcript that just gets degraded. Well, can be, but at that moment, that was the model. And there was another study from a group in Germany. They did experiments looking to natural variation, and according to them, delta was also not important, but they did experiments up to 23 degrees, and we know that delta starts to kick in at higher temperatures. So that story is completely correct, what they published, but they didn't look at exactly the right temperatures. But based on those papers, now the whole community is against it, and well, I gave it up because I think I can spend the rest of my time, of my life on figuring out what is it now. We have done targeted proteomics to see can we detect the delta protein. But I don't know if there are proteomics experts here, but you do it based on peptide detection. And the difference between beta and delta is only one large peptide, which is very difficult to catch in a proteomics assay. So we tried to do that with targeted proteomics, but we never managed to get that well running. But still, I thought I want to at least be sure that the transcript is there that we proposed that should be there. So for that reason, we did recently PacBio run, PacBio IsoSec, in which you isolate transcripts and just sequence them from beginning to end. And then you know exactly which transcripts are present. You don't use bioinformaticians to predict based on all these small Illumina fragments what are the isoforms. And if we did that for FLM, well, you can probably see already there are far more than beta and delta, and I will not go into that. We have detected, I think, 27 different isoforms, but it's very complicated. I will not go into that. But the good thing is we see a lot of beta, what we also expected, and that's with exon 2 in it. You see here an example, here, there are many. I just marked a few arrows there. Then the question is delta there? And yes, delta is there. So what the Australian people say that delta is not formed is not correct, because we see this mutually exclusive exon. We found here one, we found transcripts of delta. So delta is definitely there. So that does not say that they are wrong, because they say that it's an intron retention. And yes, that variant, you see it here, we also found. So I think the end of the story is that it's not that black and white, it's just there are all these different variants, and plants use probably both mechanisms, the delta, which is destroying part of the transcript, the one with the intron retention, and the true delta that maybe leads to a protein. And that combination of those effects gives then the nice, elegant response to ambient temperature. Yeah, so to conclude this part, FLM splicing is very important, and the FLM delta isoform, well, it is definitely there, and probably it is, especially at higher ambient temperatures, kicking in and having a role. But what I think is important to realize is that independent of what is the final fate of the transcripts, the process of alternative splicing is very important for the temperature response. Well, that triggered us also to look into crops, because in the end it's nice when you know that for Arabidopsis, but that we cannot live from. So you would like to know, can we, because that's definitely something that, for example, cauliflower growers want. They do not like that if they get a warm period during the vegetative stage, that their harvest is 2-3 weeks later, and that the product that they harvest is also of less quality. So they would like to generate temperature insensitive cauliflower variants. So for that reason, we also looked, wow, is it then maybe conserved, this pathway, and can we, based on what we know from Arabidopsis, pick the right genes and make mutants in cauliflower and come to temperature insensitive cauliflower? So what we did is, so cauliflower, I showed that when you have a low ambient temperature, you get nice curd formation. At higher ambient temperature, it's delayed and you get a less quality curd. So we did a very similar experiment where we grew cauliflower for 5 weeks at normal ambient temperature, so 22 days, 70 nights. And then we switched them to high ambient temperature for 1 week. And after 1 day, exactly 24 hours again, we took a sample for RNA-seq. After 1 week, we placed them back and grew them at the normal temperature. And the final consequence of that 1 week of heat around week 5 is enough to get a strong delay in curd initiation. So then we did again RNA-seq, differential gene expression analysis, and alternative splicing analysis. And well, hoping that it's conserved, but if we did that, we saw that the conservation at splicing level was only 6%. So it was very disappointing, because we hoped to find a big conservation, but actually the conclusion was it's not conserved at all. Well, then we, of course, we were disappointed, but we looked a little bit better into it. What is then that overlap? What are the kind of genes for which at least there is conservation? And then we saw that 10% of those 6% overlap, so 1 out of 10 is a splicing machinery gene. So it seems that the effect of temperature on the splicing machinery, that is quite conserved. But then the genes that are then selected, the flowering time genes or the growth genes that give you the final response, the developmental genes, there seems to be a variation. So based on this, we came with the model that there is maybe conservation at the mechanistic level. So alternative splicing plays a role, but it's not that the same genes are targeted. And currently, but yeah, cauliflower is slow. We have now our CRISPR mutants. We had last week, my technician came very happy that we have the 5 first seeds of a homozygous CRISPR mutant in one of the splicing factors and also in some other flowering time genes. So hopefully we can show later on by functional confirmation that really when you start knocking out these genes that you can also get a temperature insensitive cauliflower. But that's at this moment wishful thinking. Well, that was disappointing, and then you can think there is maybe no conservation at all of the finally targeted genes, but that's also not the case. And we were quite surprised there because originally I'm coming from a METS domain transcription factor lab, so I've been studying METS domain transcription factors like FLM and FLC in many different species. So we could look, is there maybe then another species where there is alternative splicing of an FLC-like gene? And to our surprise, in tomato, we found MBP8, which is very close to MAF2 in sequence. And for that, we found 2 splicing variants, one which lacks these 2 exons and the fully spliced variant. And then when we checked what is the response of the splicing on temperature, we saw very nicely that MBP8 splicing is temperature sensitive. So again, FLM-like, FLC-like transcription factor for which the splicing is temperature dependent. And while at this moment we have only correlation, but also in tomato, flowering time is affected by temperature. Then you have to think on complete different temperature regimes. So we go up to 30 degrees because tomato grows normally at warmer conditions, but then still it responds to temperature with an alternative splicing of MBP8. Well, if this is causal or just consequence, we don't know. So at that moment, we still have to generate the CRISPR mutants to show that really MBP8 is involved in this flowering time response upon a changed temperature in tomato. Yeah, so the conclusion, there seems to be at least conservation at the mechanistic level. Alternative splicing is an important mechanism. And in some species like tomato, we even found that the similar flowering time genes gets differentially alternative spliced when you change temperature. Well, I mentioned in the beginning when we were doing this genome-wide analysis that we found something like 900 genes differentially spliced. So it are quite a lot, but there is specificity. And then if you start thinking about it, how does the splicing machinery know this transcript I have to splice differently, but this transcript not? Is there something in the messenger RNA, or where is that signal originating? How can the splicing machinery go to the right loci and directly after that the pre-messenger RNA is made, give differential splicing when you have a different temperature? Well, for that we once more made use of what is known in the mammalian field. We do that quite a lot, especially if you look to underlying mechanisms. And well, this is the textbook picture how I learned splicing, and maybe many of you, so that you have a gene with the introns in it, you have transcription, and you have the pre-messenger RNA, the splicing machinery kicks in, cuts out the introns, and you get your mature messenger RNA, which is then moved into the cytoplasm and translated into a protein. Well, it's not that this is wrong, but it's not that simplistic as it is shown here. And especially in the mammalian field, they have done a lot of work that shows that splicing occurs co-transcriptionally. So it's not that first the pre-messenger RNA is formed as is shown here, and that the splicing machinery kicks in, but the splicing machinery kicks in as soon as the polymerase is releasing the messenger RNA, the splicing machinery is sitting already on top of it. Well, then you can say, well, that's a small difference, I don't care, it's just a little bit earlier. But what does this mean? That means that splicing occurs very close to the chromatin and not somewhere else in the nucleus. So, and if you think on that particular genes needs to be tagged, that they needs to be alternatively spliced, maybe that is then in the chromatin. And that would be a way to tag particular genes by having a different chromatin marks and target those for differentially splicing when the temperature is changing. Well, again, for that there was in the mammalian field already evidence. There they show that there are particular histone marks, like for example, histone 3 lysine 36 trimethylation, that directly are recognized by so-called MRG proteins, which form a direct interaction with splicing machinery genes. So due to this, depending on if this mark is there in the histones, you can get that the transcript, the different splicing machinery proteins are attracted to this particular transcript. So we were inspired by that and said, let's look to the deposition of this particular histone mark in Arabidopsis, and is this histone mark then also involved in this ambient temperature dependent alternative splicing? So for that we first looked genome-wide and we checked actually in all the expressed genes, where is this mark present? And then we see that the mark is especially present at the beginning of the gene, so quite close to the transcriptional start site. And then it's, when you go towards the end of the gene, it drops. So this is the overall pattern, and that was the same as what other people had already shown about this mark. That's in this it is different from mammalians, because in mammalians it's mainly at the end of the gene body. But in plants it's mainly at the beginning of the gene body. But then if we look to all the tier 10 genes, we see nicely this. When we look to the expressed genes, we see that there is more deposition of this mark. And that was also at the moment when we started our work, people said this mark marks genes for active transcription. So genes that have this mark are higher expressed. Well, that's definitely true. That's what you see here. If you look to all the genes and to the expressed genes, you see more deposition of this mark. But then if we took the genes which undergo temperature-dependent alternative splicing and we look to the presence of this mark, you see again an increase. So this nicely shows that the mark is mainly present at the beginning of the gene body, and especially genes with, not with, with temperature-dependent DASE are enriched in this particular mark, which could mean that this mark is involved in tagging genes and telling this gene needs to be differentially spliced. Well, this is, it's a nice way of showing a correlation, but it's just a correlation. And if you really want to prove that it's causal, then a nice way is to go to genetics. But first I would like to show you how we think that, how this mark looks and how we think that it can affect splicing. So here you see one example, and that's the PRR3 gene, which has an intron retention, which is, when you go from 16 to 25, you clearly see that it is decreasing in its intensity. And we've found it significant 1 day warm, 3 days and 5 days warm. And if you look into it, here you see that that particular intron retention. If you look then to the deposition of this histone 3 lysine 36, at 16 degrees and 25 degrees, you see exactly at this position. So this gray marks the difference. Exactly at this position of the intron, you see a difference. So in this case, it fits very well. But if you are critical, then you think maybe, but what about splicing events in the end of a gene? Because there you see hardly any deposition of this mark. But if you think on the model that the messenger RNA is of course flexible, and even at the end of the gene, because it's all flexible, it's not a rigid structure. So the deposition of the mark does not need to be exactly at the position of the splicing, because where the deposition of the mark is in the genome, there the proteins will bind that attract the splicing machinery, and maybe they affect the splicing then over there in the transcript. So we see a very nice correlation, but it's not always exactly at the position of where the splicing occurs. Well, and then the genetics comes in, and luckily for us, the deposition of this mark was already very well studied. And if you are, even if you're not so much in all these histone modifications, what you have to think of, if you want to make such an histone modification, you need a writer, and that are the SDG proteins, 8 and 26. Well, if you want to get rid of it again, you need an eraser, and a well-known eraser is Dimonyi 30. That is specifically removing this particular mark. And then you have readers, and that are these MRG proteins that recognize chromatin that contains a lot of this histone mark, and they can then directly interact with the splicing machinery and give the effect, at least in mammalians. So based on this information, we went for mutants of this particular genes and checked, are they then still temperature responsive in their flowering time response. So here once more, what we did is we grew Arabidopsis at 16 degrees, and then we switched them. So we did not do the experiment continuously 16, continuously 25. Then you see that big difference in flowering time. But we grew them first for 3-4 weeks at 16 degrees, and then we moved them to 25 degrees. But still you see a significant effect. The ones that were moved to 25 degrees get earlier flowering. And if you see then an SDG mutant, so a mutant in the writer of this histone mark, you see that this effect is almost completely gone. And the plant that you also see here from the ratio, it gets very close to 1. So independent of temperature, when the histone mark cannot be deposited, not be written in the chromatin, the plant becomes temperature insensitive. What about the eraser? When we have a mutant for the eraser, we see an increase. But if we overexpress the eraser again, which is mimicking the knockouts of the writers, we see again a decrease. So also in that respect, it fitted the data that the presence of this mark is very important for temperature-dependent splicing. And finally, the MRGs. So there are more MRG proteins, so we could get a hand on the double mutant, MRG1, MRG2. And also that, although it's not black and white, but we see in a response in that the plant becomes less responsive to temperature. So to conclude this part, normally when the histone mark is present, then under cold temperature and warm temperature, you get a difference in flowering time due to the deposition of this mark. But if you have plants in which this mark cannot be deposited because the writer is not there or the readers are not there, you get the plants independent of the temperature flower at the same moment. Well, what we are doing now in the follow-up project is trying to figure out what is now the exact cause of this effect on splicing. And there are two options, again, coming from the mammalian field. One is that it is this so-called adapter model, that this mark is recognized by these MRG proteins, and then the splicing machinery is docking on that. Another one is a so-called kinetic model, because it's very well known that depending on which kind of marks you have on the histones, the polymerase can go either faster or slower. And studies have been done with slowed down polymerases, so polymerases have small mutations. And if the polymerase goes faster or less fast, there is more time for the splicing machinery to do correct splicing. And it's known that speed of polymerase can also affect splicing. So we have now two models. One is the kinetic model. Maybe it's due to the presence of the mark, the speed of polymerase is changing, or it's the adapter model, it's the binding by the MRG proteins. One other thing that I promised you in the beginning to come back to is memory. And plants can memorize temperature. And if you think, how do plants memorize? They don't have brains. So there must be another mechanism to memorize something. And histone marks, it's very well known also for the vernalization. The cold is memorized by depositing particular histone marks in the chromatin. And here you see the same. What he does, it deposited this histone-free lysine 36 in the chromatin. So maybe the memory of temperature acts via this particular histone marks. That is also something we are currently following up in the lab to see if the memory for ambient temperature acts in the same way. Well, a lot of details at the molecular level, but I want to add one more final detail in the regulation. Again, a complete different mechanism. And that has to do with, well, we've seen now splicing, differential expression, a lot of different aspects. But if you look to this FLC clade members, especially FLC, it's known that it's regulated by so-called long non-coding RNAs. And there are many of them, cool air, cold rep, FLC, ASL. And maybe you know about them. So these are RNAs either transcribed antisense from the FLC locus or in sense, or sometimes from the promoter or intronic, and they affect the final expression of FLC. Very recently, it was also shown that for MAF4, there is an alternative antisense transcript, a long non-coding transcript, regulating its expression. So we came with the question, are also the other family members, maybe the ones which are in the ambient temperature pathway, also regulated by long non-coding RNAs? Well, the good thing was we had already that RNA-seq done 1, 3, and 5 days, and we did a de novo analysis. So I had to look at that moment a very good bioinformatician in the lab, and he could look to changes in long non-coding RNAs upon this temperature change. And well, it's the same as with the splicing. Many of them are not in tear. The same holds for the long non-coding RNAs. They are hardly in tear. And we could find actually many so-called natural antisense transcripts, long intergenic transcript, and intronic long non-coding RNAs, in total around 2,000, from which half of them were new, the other half are already annotated in some papers. So then, of course, the question comes, that's nice, but you are not going to study 2,000 different long non-coding RNAs. So you want to come up with ways to select a few that are maybe involved in your process. And for the ambient temperature change, so the change from 16 to 20, we had around 50 long non-codings that really changed their expression when you change the temperature. And among them were 11 natural antisense, 2 intergenic ones, the link, and 2 intronic ones, and 37 intergenic ones. Well, then 50 is still too much to follow up by functional studies. So we came up with all kinds of arbitrary ways to reduce this set. And what we did is we, for example, looked, are they maybe in close vicinity to flowering time genes? Because long non-codings can act in all different kinds of ways, but many of them are located close to the locus where they act on. And we also said, well, if they are important, they are probably conserved. So we also checked, is the long non-coding RNA that we have found to be temperature dependent also present in other species? And by doing that, we could bring down the set to a very few. And one of them I will show you the results of. It's called AT long non-coding 428. And this was very nicely differential between 16 and 25 degrees. And as you can see here, it's conservation. So you see that at the complete sequence level, it's you see it also in, for example, Camelina and Arabis alpina, some other Brassicaceae. But even if you go to Solanaceae, you can find small pieces of the long non-coding RNA that are still conserved. So that gave us the feeling that it's not a kind of artificial, strange transcript. There is some conservation of this transcript. So then we went to mutants, and here you see that the AT-linked mutant, when you compare again 25 to 16, you see that the effect, the difference in flowering time is reduced, and especially at low temperatures, you see where it's the highest expressed. So if you mutate it, it starts flowering earlier. So again, another level of regulation where a long non-coding RNA is probably affecting the ambient temperature response. I didn't say anything on the FLMs. The only one that we found related to these MUF genes. So we were hoping to find the similar ones like cool air and cold air for FLC, but these are definitely not there. We have not found any long non-coding for the MUFs. The only thing what we found is we found in the promoter of MUF2, there we found a long non-coding, but that we didn't follow up yet. So I have a new PhD who is studying more all these long non-codings. One thing that I have to say, it's known that the long non-codings are not all polyadenylated, and our library was made based on poly-A tailing, so it can be that we miss part of the long non-coding. So it is still possible that there are long non-codings for the MUF genes, but we missed them maybe. So based on that, we see a flowering phenotype. We renamed this long non-coding in F-link. It's an intergenic one, so flowering long intergenic non-coding RNA. Well, then back to where actually my work in the field started, as was also in the introduction. These matchbox transcription factors, they don't act on their own. They act in complexes. And with all these members, the FLC, FLM, MUFs, and then the question is, what are the interaction partners, and are they then also, is there also regulation at those genes? And for the MUFs, they can interact among each other. They don't have a lot of interaction partners. Some of these METSs interact with many of them, like the sepalata genes and proteins, but the MUFs interact specifically with SVP. Well, its name is already saying it, short vegetative phase, so also a repressor of flowering, so fitting. And we were interested in which complexes are exactly formed with SVP, and what is the role of SVP in this whole ambient temperature pathway. Well, one thing that we know about SVP, it's in this, when you knock it out, you get an early flowering plant, and when you overexpress it, you get a very late flowering plant. So it's definitely a repressor of flowering, like the FLC clade proteins. And it's not only in the ambient temperature pathway, but it's also in the vernalization pathway, it's in the gibberellin pathway, so it's clearly an integrator of flowering signals. It's in many of the different environmental factors that affect flowering, somewhere in the pathway SVP plays a role. So that makes that we said, well, SVP can interact with FLC, FLM, and so on, but is that all? And can that explain its difference in function in the ambient pathway, the vernalization pathway, and the gibberellin pathway? Or are there maybe other proteins in these complexes that give us its specificity? So to study that, we generated the line where we expressed SVP from the genomic SVP construct, so from its own promoter, tagged to GFP in the SVP mutant background. So we complemented the SVP mutant. And then we did LCMS, so quantitative proteomics. And we looked, can we identify proteins that are probably in complex with SVP? But I will not go into all the technical details, but after a lot of repetitions, also in different backgrounds, we figured out that SVP can interact as expected with FLM, FLC. So this one for the vernalization pathway, this one for the ambient temperature pathway. But we also found quite a nice set of new proteins, which were not known to interact with SVP. Some of them have a known function, like histone deacetylases for GETTR1, PDS5, but also some unc means unknown. So we also found some completely unknown ones which had no annotation at all that were interacting with SVP. So what we did then is we said, well, how to discriminate in that and which proteins to focus on. So we first altered mutants for actually for a lot of the putative interaction partners that we found, and we checked, do they have a flowering time phenotype? And well, then the one with the most strong flowering time phenotype was unknown too. And for that reason, we decided to focus on that protein more. And here you see the phenotype, and that's quite interesting because if you compare it to an SVP mutant gets early flowering, but the unknown 2 mutant gets late flowering. So it's in complex with the floral repressor, but the phenotype is opposite. So it means that it's somehow a mediator of the functioning, but counteracting the function of SVP. Well, its name that we gave it is quite complicated, PAUSP. I don't know if there are Indian people here, but the guy in our lab who figured this out, Suraj Jamde, he's from India, and he, from Sanskrit, gives it this beautiful name. I liked the translation, the one who allows it to flower or blossom, because it's late flowering, but PAUSP, for me it was a little bit difficult to pronounce, but we stick to this name because he found the gene and we thought, oh, it's a nice way of naming the gene. But if you look into PAUSP, it's a huge, it encodes for a huge protein of 784 amino acids in which you can find hardly any domains. There is a nuclear localization signal in it, and there is this glutamic acid rich area which could maybe act as a transcriptional activator. But based on its complete structure, most likely it's a chromatin remodeler. So then we checked, well, if it is interacting with SVP, where is that interaction occurring and what is it doing? So we expressed SVP in protoplast here. And although SVP is a transcription factor, if you express it on its own in protoplast, you see it mainly cytoplasmic. And then when we co-express it with PAUSP, we see that quite nicely the signal moves from the cytoplasm into the nucleus. So somehow PAUSP is one of the factors that can bring SVP into the nucleus. Also FLC and FLM can do that. When you get this heterodimer, it also efficiently goes into the nucleus. But this gives us a confirmation that probably it's a true interaction, because we have tried to confirm this interaction by yeast to hybrid studies and we never manage. It can either be because the PAUSP is a huge protein that is not expressed well in yeast, or that the interaction that we found by the mass spec is an indirect interaction, that there are other bridging proteins in between. And currently we are following this up in the lab. Well, to end up the whole story, what is then the function of PAUSP with SVP? To be honest, we don't know yet. So we are currently growing the PAUSP mutant and all other kind of mutants under different fertilization conditions, under different ambient temperature conditions to see. We found PAUSP interacting with SVP, but in which specific pathway is it needed, or is it needed in all these pathways? But there's one interesting case there. SVP, it's known, its name is also according to that, it's a flowering time regulator. But there was a study, a big transcriptomic study done years ago where they, based on transcriptomics, came with this network analysis and looked for hubs. So genes that are really central in this network for, in this case, drought tolerance and ABA signaling. And it's a plant cell paper, there they call it ACL22, but ACL22 is SVP. So SVP was found as a hub in the drought response. And we are currently also following that up because the guy that works in that, on the PAUSP protein at this moment in my lab is a Chinese. And well, the good thing of having Chinese people in the lab, they can also check Chinese literature because there is a paper about PAUSP, 1813 it's mentioned in this paper. And well, maybe you can read it. I have no idea what is all written there, but he figured out that people have already been working on PAUSP and they could show that the PAUSP mutant is having an effect on drought resistance and on ABA sensitivity, which would fit perfectly with the interaction of PAUSP and SVP and being a key player also in the drought response. And that is something we are currently following up. So to summarize this and to conclude that, plants have, as you have seen, a tremendous capacity to adapt their growth and development to the environment. Well, temperature is definitely a very important signal in that, and plants make use of it for timing of reproductive decisions in order to guarantee a reproductive success. Members of the matchbox transcription factor family, they are key regulators of the ambient temperature dependent flowering time control. So not only the FLC clade members, but also SVP. And I hope I convinced you that alternative gene splicing is very important and can act as a direct thermosensor to make plants respond in a proper way to temperature changes. And with that, I'd like to end, but first I would like to mention a few people from the lab. So this is from the own group. Alicia Pagora was a postdoc. She's now in the lab of George Coupland at the Max Planck in Cologne. And she did the majority of the work on the histone marks and the link between histone marks and splicing. All the initial splicing work was done by Leonie Verhagen, PhD in my lab. She's now in the lab of François Passy in France. Suraj did the work on SVP. Miao Bai is following that up. And I'm still a member partly for my time in of Gerco Angenet, and well, he was my PhD supervisor, and he's really an expert in all this METS work. So he, as supervisor, was involved in that. And for the Cauliflower work, we collaborate with Johan Bugger and Guusje Bonnema. All the bioinformatics that I show, because that you have to realize that all this de novo analysis, even in Arabidopsis, but also in Cauliflower, is really challenging. Look to these long non-codings, to the splicing. And in that, I was really happy that I had Eduard Severing, but unfortunately, he also left to George's lab. All my good people go to George Coupland. But I'm really happy, and still once in a while we collaborate. And finally, the collaborators are, George is also a very good collaborator, Brandon Davies, Marcus Smith, Pavel, and Marcel Proveniers of Utrecht University. And of course, I have to thank our financiers, which include also quite some breeding companies in the Netherlands, Beo, Syngenta, and Rijkswaan. And I really appreciate their support in translating all this knowledge to crops such as cauliflower. Thank you for your attention, and I'm open for questions. Yeah.
So you talked about one of the LncRNAs, LncRNAs that had an effect. Is that the only one you checked, or the only one that had a phenotype?
It's a good question that I can be honest about. I think we have it somewhere written in the paper. It's just out end of last year. We checked 7 in total, and this was the only one that gave a flowering time phenotype. We had 2 other ones that give a growth phenotype. This is also interesting because temperature affects also growth. But I have to be honest, those were not characterized very much in detail. And it was the work of Alicia. She left then. It was the end of her postdoc. So yeah, we had to stop there. But now I have a new PhD, and she will focus more into it. Also, we will do more analysis on that 50 to make a better selection. And for that, also focus mainly on those that are going back into the nucleus, those long non-codons, or stay in the nucleus and are functioning there.
I think part of the interest in the cauliflower funders was that there's a desire to create varieties that have various predictable flowering times.
Exactly.
And probably the hope was to do that by marker-assisted selection, but I've not seen how one would do marker-assisted selection for this particular thing.
Yeah, that's a very good point, and that's also, as you have seen, we have also not solved it yet, but actually that's already, I think, 4 or 5 years ago. Then these breeders came to us because they work together with a lab in Germany. There are also some publications where they use classical QTL mapping and so on to try to identify what is ongoing. And there are some temperature insensitive cauliflower varieties. So then you think, well, it's easy. You just cross them and you do the mapping and you're done. Well, they have used this a lot, and well, in a few generations it stays, but then it disappears. And that's why they said we need more insight in what are now the underlying mechanisms. And then we thought, well, in Arabidopsis it's easy. We will do the same in. We completely underestimated how difficult that will be. But together with the companies, we still have a project running. Actually, when I'm back the day after, we have a meeting with them, a progress meeting. It's almost over, and we are thinking now also on a new project where we will, because it's not only the switch of the flowering that makes them, but also afterwards, temperature has a strong effect on the whole inflorescence development. And we till now only look to the initial induction, but we hope that they are willing to also look with us more to what is happening after that. Yeah.
Is it possible that the large number of RNA splicing genes that you are seeing in the RNA is because of feedback loop? Did you check any of those genes just before 24 hours, like just by real time?
There are all kind of loops, and definitely we decided to look only after 24 hours because of we knew that we didn't want to have also the clock. It's already complicated enough, and if you have then also the circadian rhythm involved. But we know for FLM we have been looking after 2 hours, 4 hours, and yeah, you see a lot happening, very fast responses, and maybe even at 24 hours there are already feedback loops kicking in, and so it is complicated. Yeah, and we had to make this selection 24 hours and then and so on. But definitely there are things happening much faster, and it's a very complex regulatory mechanism. Yeah. Good point.
So
Yeah.
you showed that these genes involved in HSV sort of six resolution, they all have flowering effect.
Yeah.
But do you know, are they affecting through the same target gene or?
That's a good point, and in that I have to be honest. So when you modify this histone 3 lysine 36 primate all this, they have a flowering time effect. For the rest, you would think maybe they would be very pleiotropic, and some of the histone marks are that you get this very small dwarf plants. But when you have mutants in this mark, that's not the case. Although you also see a little bit in vegetative growth an effect. But which flowering time genes are exactly affected? FLM is definitely affected, but the ratio that you see in FLM splicing alone is not explaining the effect on flowering time. So it's probably a combination, and that is also what I showed, also PRR3, PRR5, 7. It's a combination of all the genes that gets differentially spliced. So it's also not that when you remove the mark that you have no splicing at all, but it sometimes is that the isoform ratio is swapping in the other direction. And it's very complicated. On some genes, it's increasing the splicing. Other genes, it's decreasing the splicing. And we are far from understanding that. Yeah, so there's a lot to do there still. Very good point.
Interested how stable the temperature memory is. So if the higher temperature memory move them back to a cooler temperature, how many of those genes actually remain expressed in that?
Yeah, I will repeat it. So the question is how stable this temperature memory is, so if you change it back, which of the genes remain their ratio? We only have done in that respect because we were always focusing on FLM, and there's this work from Steve Penfield, and I must say I trust his data, but I know that there are a lot of labs having difficulties in reproducing it, also because he's using Columbia, and Columbia has hardly any seed dormancy, so you have to do all kind of tricks to see undormancy effect, and if you have a slightly variation in your experiments, it's already gone. So we had quite a big problem in that, but we have seen for FLM, we have looked, and for FLM, we have seen in only in a few pilot experiments, and then it stopped. So it's for the new PhD, we'll follow that up. We have seen that it's memorized, but how general that is, we don't know. And if these histone marks are involved in that, I also don't know. And I thought of looking to that, but I decided then, if I want to do this thoroughly, I can better do it with a new PhD. And what I will do is select first an Arabidopsis succession that has a very nice temperature-dependent flowering time response and dormancy response. And in that, I will, nowadays with CRISPR, you can easily do that, remake the SDG mutant, all the mutants, the FLM mutant, and then see how it is, because I don't want to have a PhD struggling again with this very difficult Columbia, which is hardly dormant. And then we also have to see, it's still a theory and very hypothetical, so it's a good point. We believe that it's like that, but we don't have a lot of evidence yet. Yeah, very good point. Yeah.
This has been a production of Cornell University, on the web at cornell.edu.